Good morning, ma'am. Good morning, everyone. My seminar topic in molecular biology is regulation of gene expression, that is Unit 5. The main topics under discussion are antitermination N protein nut site and ARA operon of E. coli. The content of discussion in this seminar PPT are the gene expression, operon, positive gene regulation and negative gene regulation, regulation by attenuation, antitermination, N protein nut site. Gene expression. To know what is gene expression, we need to know what is gene. We are all aware that gene is a basic physical and functional unit of heredity and it is made up of DNA. Some genes act as instructions to make molecule called protein, and gene expression is a process by which information from a gene is used in the synthesis of functional gene product that enables it to produce end product protein or non-coding RNA and ultimately affect a phenotype as final effect. Examples of gene expression can be control of insulin expression, so it gives a signal for blood glucose regulation, and cyclin expression levels control progression through eukaryotic cell cycle. The gene expression can be measured by identifying viral infection of the cell and even by protein quantification method or mRNA quantification method. We can even find it by checking the bacterium resistant to antibiotics, that is penicillin. We have gene expression databases like Gene Expression Omnibus, that is GEO at NCBI, and Expression Atlas at EBI. The gene expression is used by all the known life, that is eukaryotes, prokaryotes, viruses, to generate macromolecular machinery for life. It is summarized in Central Dogma Molecular Biology. We all know that central dogma is a stepwise process to produce from DNA to a protein. We have the steps like DNA replication, transcription, translation, and reverse transcription in it. We all know that DNA replication is a process by which DNA makes a copy of itself during cell division, and transcription is a process by which information of a strand of DNA is copied into a new mRNA, that is messenger RNA, and enzyme used as RNA polymerase. And reverse transcription is a process in which reverse transcriptase enzyme is used to generate complementary DNA, that is cDNA, from RNA template. And translation is a process by which RNA is used to produce protein. It results in the formation of polypeptide, which is regulated at various levels. All steps in gene expression process may be modulated or regulated, including the transcription, translation, RNA splicing, post-translational modification of proteins. It results in the formation of polypeptide, as discussed earlier. And regulation of gene expression gives control over timing, location, and amount of given gene product present in a cell. It is the basis for cellular differentiation, development, and morphologies of any organism. In genes like constitutive gene, housekeeping gene, facultative, and indivisible gene are involved in it. Jacob and Monod in 1961, on the basis of their study on inducible system for synthesis of beta-galactosidase enzyme in E. coli, proposed a model, that is to explain the induction or repression of enzyme synthesis. And the scheme produced was called as Operon model. Operon is a functional unit of DNA containing cluster of genes under the control of single promoter. The term operon derived from verb to operate. It occurs primarily in prokaryotes and in some eukaryotes. It contains one or more structural genes which are generally transcribed into one polycystic mRNA. The operon is made up of three basic components, that is promoter, operator, and structural genes. Promoter is a nucleotide sequence that enables gene to be transcribed, and operator is a segment of DNA to which repressor bind, whereas structural gene is a co-regulated by operon. It directs the synthesis of cellular protein through mRNA and determine sequence of amino acid in protein and synthesize the lactose lac of bacterium. It is a small segment of circular DNA molecule which constitute genetic material on bacterium. In this diagram, we can understand the structure of operon. Here, this is RNA polymerase, and this is a repressor protein, and this the one which is showing in white curve plus, that is a lactase, and these are the structural genes lag Z, lag Y, and lag A, and this is a promoter operator. The gene is initially turned off. In this diagram, the gene is initially turned off. There is no lactase to inhibit the repressor. So the repressor binds to operator, which obstructs the RNA polymerase from binding to the promoter and making lactase. And here the gene is turned on. That is, the lactose is inhibiting the repressor and allowing the RNA polymerase to bind with promoter and express the genes which synthesize lactose until there is none to bind to the repressor. Repressor will be then bind to operator to stop the manufacture of lactase. This lactase, upon addition of beta galactosidase enzyme, give rise to galactose and glucose. Operon, that is, they have two negative gene regulation and positive gene regulation. Negative gene regulation, that is lack operon in E. coli, and positive gene regulation, that is ARA operon in E. coli. The lactose operon of E. coli encodes the enzyme beta galactosidase, which hydrolyzes lactose into galactose and glucose, as we observed in the previous slide. And the protein that inhibits transcription of lac operon is a tetramer with 4 identical subunits called lac repressor. It is active only when it is in the tetramer form. The encoded this lac repressor encoded by lac-I gene located upstream of lac operon. And in positive gene regulation, that is E. coli, that is arabinose operon, the ARA operon, the C protein bind arabinose and undergo conformational changes that enables it to also bind the ARA O2 and ARA I2 site. This we can will understand in the coming slides. The results in the generation of different DNA loops is formed by interaction of C protein bound to the ARA1 and O2 site. The formation of this loop stimulates transcription of ARA-C gene, resulting in additional C protein synthesis. The C protein autoregulates its own synthesis. This was a short description of what is positive gene regulation. Now, the main topic of discussion is that is E. coli ARA operon. It was first studied by Ellis L. Egglesberg. The arabinose is 5-carbon sugar and a constituent of some plant cell walls as a source of carbon and energy. Since availability of arabinose is sporadic, one would expect the ability to utilize the sugar to be inducible. Arabinose is an operon can be regulated both positively as well as negatively. The structure of operon. The ARA operon is composed of structural genes and regulatory regions, that is ARA-C, including the operator region, that is ARA-O1, and ARA-O2, that is operator 1 and operator 2, and initiator region, that is ARA-I1 and I2. Sorry, there's a slight correction here, I1 and I2. The structural genes ARA-B, ARA-A, and ARA-D, collectively known as ARA-BAT. They encode for metabolic enzyme. These are the structural genes. And ARA-B codes for ribonucleokinase, ARA-A and isomerase, ARA-D and epimerase are produced by these genes. Additional genes, that is ARA-E, ARA-F, and ARA-G, ARA-H, are present in this arabinose operon. There is also a cap binding site where CAMP complex binds to facilitate catabolite repression and results in positive gene regulation of ARA-BAD. The regulatory genes of ARA-C, that is C protein, is located upstream of the arabinose operon and encodes for ARA-C protein. Both ARA-BAD and ARA-C, arabinose C, are transcribed in opposite directions from ARA-BAD promoter and ARA-C promoter respectively. Here in this diagram, you can see the different sections made. That is ARA-C, that is regulatory gene, and ARA-O2, that is operator 2, and this is promoter for ARA-C, ARA-O1, operator 1, CAP, and ARA-I1 and I2, that is initiators regions, and then structural genes, that is ARA-B, ARA-A, and ARA-C. These are encoded by the enzyme ribokinase, isomerase, and epimerase. The operon is inducible and inducer is arabinose itself. That is in this wild type of operon, ARA-BAD mRNA is made only when arabinose is present. We understood that there are two types of controls, that is positive control of ARA-C operon and negative control of ARA-C operon. The components of arabinose operon are the following listed below. That is gene C, B, A, D, O, operator 1L, that is left, and operator OR, right, CRP, I1, that is initiator 1, initiator 2, promoter C, and promoter ARA-BAD. Each one has the following base pair size, that is C gene has 1125 base pairs, and the protein is a repressor, and its molecular weight is 42.2 kilodalton. It functions as repressors and ARA-BAD gene. The gene B has 1842 base pairs. The protein is ribulose kinase, and the molecular weight is 66.49 kilodalton. And gene A, that is 1500 base pair in size, and the protein is isomerase. Its molecular weight is 55.8 kilodalton. And D, gene D, that is size is 772, protein RUMP epimerase, and the molecular weight is 32 kilodalton. The operator 1 has two half sites, that is one at the right to promoter, that functions that is located to the right to the promoter, and then operator 2, that is one has another half site 250 from the promoter start, and then located at minus 200 to 250 in the C gene itself. The CRP has two half sites again, and the proteins are 120 to 160 in between this base space, and it is located between operator 1 and initiator 1 site, and initiator 1 has 17 base pair, that is half of it, but the protein is at approximately at 40, and then they act as inducers at inducer sites, and inducer 2 as well as has 17 base pair, and the protein is at 60, and then it function as inducer site. Promoter C, the function is not known, component function not known. Promoter ARA-BAD is located at the left of the ARA-BAD cluster. Positive gene regulation by ARA-C protein. The expression of ARA-BAD operon is activated in the presence of arabinose. When arabinose is present, both ARA-C and CAP work together and function as activators. It can follow 3 to 4 ways. The one which I'll be discussing mainly is ARA-C. It can even follow by CAMP pathway, etc. The ARA-C function as a homodimer, which can control transcription of ARA-BAD through interaction with the operator and the initiator region. Each ARA monomer is composed of 2 domains, including a DNA binding domain and a dimerization domain. ARA-C undergoes conformational changes when arabinose binds to the dimerization domain. As a result, the ARA-C operon arabinose complex falls off from ARA-O2 and breaks DNA loop. One of the monomer binds to ARA-I1, the remaining monomer binds to I2. One of the ARA-C monomer places near to the ARA-BAD promoter in this configuration, which helps to recruit RNA polymerase to the promoter. One of the ARA-C monomer places near to the ARA-BAD promoter in this configuration, which helps to recruit RNA polymerase to the promoter. In this diagram, we can understand how ARA protein is acting as an activator as well as the repressor. A ARA gene encodes as an activator protein that, when bound to arabinose, activates transcription of the ARA operon, perhaps by helping RNA polymerase bind to promoter located in the ARA-I region. And how ARA-C protein acting as a repressor? It is actually a dimeric ARA-C which act as repressor. One monomer bind to operator of ARA-BAD gene, that is ARA-O2, and another monomer bind to distant DNA half site, that is known as ARA-I1, that is initiator 1. This leads to formation of a DNA loop. Regulation by attenuation. Attenuation regulates the termination of transcription as a function of tryptophan concentration. At low levels of tryptophan, full length of mRNA is made, whereas at high levels of transcription of TRP operon is prematurely halted. Attenuation works by coupling with transcription and translation. The TRP operon can be and his operon comes under regulation of attenuation. That is, TRP operon it codes 5 structural genes involved in tryptophan biosynthesis, whereas his operon has leader which encodes a peptide with 7 successive histidine codons and 1 transcript encoding all 5 synthesized using single promoter, that is PTRP, and operator OTRP site. Whereas in his operon, his operon has no repressor. Operator regulation and attenuation forms only by the mechanism of feedback control. Antitermination. Antitermination, we all know that transcription, translation, DNA replication has a stage called termination. That is where the process ends. The transcription termination is the process where nascent RNA is released from the complex with RNA polymerase and the DNA template. Whereas in translation, the termination happens when a stop codon in the mRNA, that is UAA, UAG, or UGA, enter the A site of tRNA. And then termination of DNA replication occurs when 2 replication forks meet on the same stretch of DNA. Whereas antitermination in prokaryotic cell is aid to fix premature termination of RNA synthesis during transcription of RNA. It occurs when RNA polymerase ignores the termination signal and continues elongating its transcript until second signal is reached. Anti-termination is used by some phages to regulate progression from one stage of gene expression to the next. Anti-termination was discovered in bacteriophage infection first. The lambda phage is used at two stages of phage expression, and the anti-termination protein produced at each stage is specific for particular transcription units that are expressed at that stage. N protein and nut site. This anti-termination takes place in N protein or at nut site. The anti-termination activity of protein N is highly specific, but the anti-termination event is not determined by the terminators, that is TL1 and TR1. The recognition site needed for anti-termination lies upstream in transcription unit. The recognition site required for protein N action is called nut site for N utilization. The site responsible for determining leftward and rightward anti-termination are described as nut L and nut G respectively. Anti-termination protein of bacteriophages and other phages which play a key role in the entry stage of infection. During early phases, any two genes, that is N and CRO, are transcribed by transcription of the DNA in the opposite direction. The N protein binds to the sites on the DNA, preventing row-dependent termination and allow transcription of genes. The regulation of the life cycle of E. coli phages, lambda, is controlled by modulating transcription termination. The N protein interacts with the sites in the DNA and inhibits the normal termination, the phenomenon that is called as anti-termination. Early in both lytic and lysogenic pathway of lambda transcription initiated from the promoter PI and yield a short transcription called LI. This transcription includes a gene N whose product is anti-termination protein. Transcription by unmodified post-RNA polymerase stops shortly after the young genes because the polymerase encounter the normal transcription termination sequence TL1. Following the transcription of the N genes, translation of LI occurs and the N gene product is synthesized. The N gene product binds to short RNA sequence that is transcribed from the DNA located nearby called N utilization site or NUT L. In this diagram, we can easily understand how the N protein and anti-termination is taking place. In the diagram A, protein N binds to RNA polymerase at NUT site. That is, the transcription include a gene N whose product anti-termination protein. Here and then in this diagram B, RNA polymerase continues post-terminators into delayed early genes. Here the protein N binds to the RNA polymerase and the enzyme continues its function. The N gene product binds to short sequence of RNA transcribed from DNA located nearby, that is near NUT site and utilization site. Here, my topic of seminar comes to an end, and today in this seminar, I discuss mainly on anti-termination, that is N protein and NUT site, and ARA Operon. Thank you.